Wait. Okay. Can you see my screen?
Yes, we can.
Okay. So my name is Carlos Pérez López. I am a visitor student from Barcelona. And as some of you don't know me, I have prepared a couple of slides for introducing me and for introducing the topic of my presentation. So, wait. Okay. So about myself, as I said, I am from Spain. I'm working in Barcelona, but actually I'm not from Barcelona. I'm from a small village among Barcelona, Madrid, and Valencia. And my city or my village is called Teruel, and it's in the region of Spain with the lowest population density. But anyway, if you visit Spain, you have to visit my village. I encourage you because we have the biggest paleontologic park in Europe, for example, and we have a kind of art in that you can only find in that region. It's called mudéjar. It's from the 12th century, and it's a mix of Christian and Muslim art. So I think it's very interesting. And next season you can watch football games or soccer game for you in the third division of Spain. Just an extra fact. Okay, and about my PhD, as I said, I am working in Barcelona. I am doing a PhD in bioinformatics. I start in 2020, and it's supposed I will defend in 2024. I am working on the development of bioinformatics and chemometrics tools for chemical compound resolution, quantification, and identification in complex mixture, mainly in proteomics and metabolomics mixtures. And I am working at the Institute of Environmental Assessment and Water Research. This is the environmental research center of the Spanish government. And I am working in the Department of Environmental Chemistry. In this department, our studies are focused on water, air, soil, crops, and biota. And in my case, I am working with water and biota samples. And today I want to talk to you about the water, more specifically about the wastewater. And now I ask you a question, which kind of information do you think that we can obtain from wastewater? So with my presentation, I want to answer this question and give you an idea about the importance of the wastewater and wastewater treatment plants in the study of exposomics and human health. So, first of all is which kind of molecules can we find out in wastewater? I have divided the total of these molecules in two groups. The first one is small molecules. Spoiler, the second one is large molecules. But in this first group, I have enclosed the pharmaceuticals, illegal drugs, pesticides, personal care products, alcohol, tobacco, among others. And in the second group, large molecules, and nucleic acid chains and proteins. I will give you a couple of examples of the information that we can obtain from these molecules. So let's start with the small molecules. In Spain, some years ago, one newspaper published this article. I'm sorry because the articles are in Spanish, but voila. Barcelona consumes 70,000 doses of cocaine each day. But I don't know if you, but at least I ask myself, how can they calculate this number with an illegal business and illegal consumption? So with the help of wastewater is easier because, for example, in this paper, the researchers assessed the drug use in Barcelona during 5 years using wastewater-based epidemiology. And they conclude that alcohol, cannabis, and cocaine were most consumed drugs. But the important point and the interesting thing was the third point, that drug use increased from 2011 to 2015, contrary to the national official data. And here is the difficulty of the calculation of these numbers in illegal business or illegal sales and consumption. So I think wastewater can help with this monitoring. So another example is the pharmaceuticals. I think pharmaceuticals, the incorrect use of pharmaceuticals is a social problem. And here we have another newspaper, Spain increasingly medicated. And this is true, but how can we monitorize that increasement? So here, these researchers compared the data in wastewater with the data in official reports, and the measured consumption of pharmaceuticals in wastewater is higher than the prescription-based consumption. Pharmaceuticals not included in prescription data report highest differences. This means that we use pharmaceuticals in a horrible way, and if we don't need a prescription, we consume everything. So, and the other point is the demonstration of wastewater-based epidemiology as a good approach to estimate total consumption. So, after these two examples, I want to move on large molecules. I'm going to start with nucleic acid chains, RNA. If I talk about RNA in wastewater, I have to talk about COVID-19, about the pandemic. And as you know, during the pandemic, a lot of research fields joined to fight against that situation. So wastewater-based epidemiology was not an exception. And here, this newspaper highlighted the contribution of wastewater in the fight against COVID-19. So in this paper, for example, the researchers compared the data of the RNA and in different wastewater treatment plants during 2 different waves, and compare the results with the official data, with the hospitalizations and the infections. And they conclude that wastewater loads are congruent with estimation of active cases within the sewer seed. In addition, and for me this is the point, wastewater saw good correlation with the number of cases that would be diagnosed from 0 to 7 days after sampling. So can we use the wastewater as a predictor, a kind of predictor to prepare the hospitals, to prepare everything? I think this is very interesting, but it is not perfect, and it has the drawback that the smaller wastewater treatment plants had problems, and the quantification was not so accurate when the RNA was in low levels. So, but anyway, I think this has a lot of potential. And finally, I want to talk about proteins, about a field called environmental proteomics. So I'm going to start with this paper published by my supervisor, and I see this paper not as a great result. I see like a test for testing the information that we can obtain from proteins in wastewater treatment plants and in wastewater in general. So for this study, wastewater samples were collected from 3 different different sites in the wastewater treatment plant: the influent, the anoxic reactor, and the effluent. And a total of 690 proteins from bacterial, plants, and animals, including humans, were identified, showing different proteome profiles in different sites. Here is a table with the most represented proteins in PSMs, and there are more proteins, obviously. Here, no, there are not almost 700, but for me, the important conclusion of this paper was that they could identify disease biomarkers related with inflammation, renal tubular function, and a lot of defensins, interlectins, or even immunoglobulins. And some of them are among the most represented proteins. Here they are highlighted. But okay, the question now is, has environmental proteomics in wastewater practical application? Because I gave you some examples in the other molecules, but what about environmental proteomics? I'm gonna change a little bit the topic, but we will be back soon. And I want to comment the last article: rats take over the gardens of Palau Robert in Barcelona. But how is the rat population monitored? Right now, we are using mainly traps and camera, but I think these techniques are not as accurate as it should be, because it is increasingly difficult to catch them with traps, and it is impossible or almost impossible to know if the rat seen on the camera is the same one or a different one than a rat you saw maybe 2 hours ago, for example. So I think we need a more accurate technique. So in this point, in Barcelona, we started another study with environmental proteomics, comparing 10 wastewater treatment plants influence, the influent water, and we obtained this that is very interesting. We were able to differentiate the human and murine pancreatic amylases, and the detection of pancreatic amylases from rodent in wastewater indicates the presence of live animals, and its relative quantification to human amylase could allow to monitor the increase or decrease of rodent phase in these samples. Therefore, wastewater can be a great source for rodent detection in cities to create a rodent map. Until now, this is only theoretical, but I think it could be a huge improvement in plague control. And to conclude, the take-home message for you, I want to you remember that the study of wastewater provides a huge amount of information of the health and habits of the population. The accurate knowledge can be obtained using the study of wastewater, obviously together to the official reports, and that the wastewater can help in the monitorization of COVID-19 or maybe other disease, and to control the plagues. And the most important point is that humans are reconquering Barcelona from rats, so you can visit Barcelona. And thank you to my group in Barcelona, Environmental Proteomics, Chemiometric Groups, and also all of you for giving me the opportunity of staying here. And thank you so much.
Great, thanks very much.